In the summer of 1953, a woman named Edith sat at a kitchen table in Birmingham, Alabama, watching her two daughters eat breakfast. The older one, Clara, was 9 years old, dark-skinned like her father, with tight black curls and deep brown eyes. The younger one, Ruth, was 7, and she was pale. Not mixed-race pale, not light brown pale, white pale. Blue-veined, freckled, sandy-haired pale, Ruth looked like she had come from an entirely different family. She had, in a sense, come from an entirely different lottery. Same mother, same father, same bloodline running back through both sides, and yet nature had dealt these two sisters a completely different hand of genes, producing two children who, under the laws of Birmingham, Alabama in 1953, would be assigned to different schools, different water fountains, different futures, based entirely on the luck of which melanin variants each of them had drawn at conception. That morning, Edith was not thinking about genetics. She was thinking about the walk to school and about the two different routes she would need to take her daughters to reach two different buildings, and about the way white women on the street looked at Ruth and then looked at Edith and then looked away with something complicated moving behind their eyes. She was thinking about the conversation she had already had with Clara, age 9, about why her sister got a different lunchbox, why her sister sat in a different classroom, why the world outside their front door seemed to be organized around a fact that made no sense inside it. She was thinking about how to explain to a 9-year-old that her government had decided, in careful legal language, that her little sister was something she was not. This is a story about what a few genetic variants can do to a family, not what they do biologically. What they do when a society decides that biology means something it has never actually meant, and then builds laws, institutions, and inherited cruelties around that misreading generation after generation, until the misreading becomes ordinary, becomes the water people swim in, becomes something a mother in Birmingham has to plan her morning walk around. The science of skin color is one of the most thoroughly studied areas of human genetics, and it tells a story that is almost perfectly opposite to the story that societies have built around it. Human skin color is controlled by a single pigment called melanin, produced in specialized cells called melanocytes that sit in the deepest layer of the outer skin. Every human being born on Earth, with the exception of those carrying rare albinism conditions that disrupt melanin production entirely, makes melanin. The difference between the darkest human skin on the planet and the palest is not a difference in pigment type. It is a difference in quantity and distribution of the same biological substance. That is the entirety of it. The spectrum that divided Clara from Ruth at the breakfast table, that put Ruth in one school and Clara in another, that reshaped an entire family's life, is a spectrum of more melanin and less melanin, regulated by variants scattered across more than 100 genes, reshuffled in every single pregnancy since the beginning of human reproduction, producing an outcome that no parent can predict and no government can map onto a stable boundary. To understand how those genes work, you need to understand that melanin itself is not one thing. It comes in two primary forms, and the balance between them shapes most of what you see on another person's face. The first form is eumelanin, a brown-black polymer responsible for the deepest skin tones, the darkest hair, the most richly pigmented eyes. The second form is pheomelanin, a yellowish-red polymer that produces the warm tones in red hair, the golden undertones in many lighter complexions, the faint blush visible in very pale skin. What your melanocytes produce, how much of each form, and how efficiently they package and transfer that melanin into surrounding skin cells, is what determines your skin tone. A person with very dark skin is producing large quantities of eumelanin in densely packed, efficiently distributed melanosomes. A person with Ruth's coloring is producing far less total melanin, with pheomelanin making up a larger relative share. Every shade between those 2 points is a different configuration of the same system, not a different category, not a different kind of human, just a different output of the same biological process, running on different combinations of inherited variants across a genome that does not know what century it is living in, and does not care what laws the people carrying it have written. For most of human history, researchers believed skin color was controlled by a small handful of genes, perhaps 4 or 5, perhaps a dozen. As genomic tools became more powerful, that estimate grew. A landmark 2017 study led by geneticist Sarah Tishkoff at the University of Pennsylvania measured skin reflectance objectively, using a color meter pressed against the inner arm across more than 2,000 people from ethnically diverse African populations, and then examined more than 4 million genetic variants across their genomes. The study found 4 key gene regions strongly associated with skin color variation in Africa. One of these, a gene called MFSD12, had never before been linked to pigmentation at all. When researchers blocked its expression in cell culture, eumelanin production increased. When they knocked it out in zebrafish, yellow pigment cells disappeared entirely. The gene was operating on the melanin system in ways that the existing scientific literature had missed entirely. A 2024 genome-wide screen published in a peer-reviewed journal identified 169 functionally diverse genes converging on melanin production pathways, of which 135 were previously unknown associations. After decades of intensive research, scientists are still finding genes that shape the trait that Birmingham, Alabama decided it understood well enough to use as the basis of an entire legal architecture. Tishkov's study revealed something else that carries its own weight. The study found the darkest skin in Nilo-Saharan pastoralist populations from eastern Africa and the lightest skin in sand hunter-gatherer populations from southern Africa. The full range of human skin color, from the very darkest to the very lightest, exists within the African continent alone. This single finding makes the concept of skin color as a marker of racial category not just socially constructed, but biologically incoherent at its foundation. The richest melanin variation on the planet does not exist between racial groups. It exists within the population that every other population on Earth descends from, because every human alive today traces their ancestry back to Africa. And that means every gene variant controlling melanin levels in every body on Earth originally came from the same place. Ruth's pale skin and Clara's dark skin were not a meeting of different races. They were a reshuffling of variants from the same ancient human genetic library, expressing themselves differently in two sisters born 2 years apart. The evolutionary reason skin color varies at all is a story about sunlight, and about a trade-off that the human body has been navigating since our species first left the environments near the equator where it evolved. Ultraviolet radiation from the sun does two important and opposite things to the human body at the same time. It damages DNA, breaking strands apart, and generating mutations that can, over time, lead to cancer. It also breaks down folate, a B vitamin so critical to fetal neural tube development that its deficiency during early pregnancy is directly and strongly linked to severe birth defects, including spina bifida, a condition that kills or disables children before they ever learn to walk. Melanin absorbs ultraviolet radiation efficiently, sitting in the outer skin layers like a natural shield. More melanin means more UV protection. Darker skin, near the equator, where UV radiation is intense year-round, protects DNA and preserves folate. Natural selection maintained it there across hundreds of thousands of years, because the women whose darker skin preserved their folate were more likely to carry healthy pregnancies to term, and their children inherited the genes that had kept them alive. But ultraviolet radiation also does something essential. It triggers the synthesis of vitamin D inside the skin, and vitamin D is not optional. It regulates calcium absorption, immune function, cell division, and dozens of other processes without which the body begins to fail. In northern latitudes far from the equator, where sunlight arrives at a low angle and passes through a thick column of atmosphere before reaching the ground, UV radiation is weak, particularly in winter, and particularly for people with dark skin, whose melanin blocks the small amount of UV that does arrive before it can trigger vitamin D production. Natural selection in these environments, over many generations, favored lighter skin, because every reduction in melanin that cost a little UV protection paid for itself many times over in vitamin D production in sunlight-scarce conditions. The gradient of skin color across the globe, from darkest near the equator to lightest at the highest latitudes, is not the result of different groups of people being different kinds of people. It is the result of natural selection solving the same trade-off: more UV protection versus more vitamin D, differently depending on where people happen to live. Critically, this solution was arrived at independently through different genetic routes on different continents, because that is how evolution works when it finds the same problem in different places. Lighter skin in European populations developed primarily through variants in genes called SLC24A5, SLC45A2, and TYR. Lighter skin in East Asian populations developed through largely different variants, primarily in OCA2 and other genes distinct from the European set. Same phenotype, different genetic architecture, same evolutionary pressure. Two completely independent solutions arrived at separately on opposite sides of the world. This is called convergent evolution, and it is one of the clearest demonstrations in all of human biology that skin color is a response to an environment, not a marker of a kind. When natural selection needs lighter skin in a high-latitude environment, it builds it from whatever genetic material is already present in that population. It does not care which genes it uses. It cares only about the outcome. Clara and Ruth's father understood more of this than most men of his generation were given credit for understanding. He had not studied genetics. He was a carpenter. But he had watched his two daughters enter the world, and he had thought, carefully and for a long time, about what he was looking at. He had noticed that his own mother had been lighter than him, and that her mother, his grandmother, had been lighter still. A woman born in Georgia in 1889, whose own parentage was a subject that was never discussed in the family, the silence around it carrying its own kind of information. He understood, without being able to name the mechanism, that skin color moved across generations not in a straight line but in a scatter, jumping lighter and darker in ways that followed no simple logic, because the genes driving it were not one gene but dozens, inherited in separate pieces, assembling themselves into a new configuration in each child the way cards assemble into a new hand from the same shuffled deck. What he could not have known was how precise the science would eventually become in describing exactly what he had observed. A 2013 longitudinal study, published in a peer-reviewed family science journal using data from 767 African-American families, documented that families displayed measurably different parenting behavior toward their own children based on skin tone. The same parents, different treatment. Lighter-skinned daughters received higher quality parenting on measured indices than their darker-skinned sisters. Darker-skinned sons received more racially protective socialization than their lighter-skinned brothers, a difference the researchers interpreted as parents unconsciously preparing darker children for a world they understood, from experience, would be harder on them. This is not a story about bad parents. It is a story about a pressure so pervasive and so old that it had worked its way inside the most private space in human life, the space between a parent and a child, and was operating there invisibly, shaping lives before those children were old enough to understand why. Research from the National Bureau of Economic Research, using full-count United States Census data from the late 19th and early 20th centuries, found that skin tone was a stronger predictor of occupational status and income than parental socioeconomic background itself. Darker-skinned respondents were roughly twice as likely to report experiencing discrimination in a given month compared to those with lighter complexions. A Duke Law Review analysis, drawing on decades of documented data, found darker skin associated with longer prison sentences for identical crimes, lower wages for men, lower marriage rates for women, and lower perceived intelligence in evaluator studies. All of this driven by a trait that is, at the molecular level, a variation in the output of the same cellular machinery running in every human body on Earth. In Birmingham in 1953, Edith did not need academic journals to tell her any of this. She lived it in the morning walk. She lived it in the way Ruth moved through the world, effortlessly, invisibly, the world bending gently toward her, in ways that Clara had already begun to notice and could not yet fully articulate. She lived it in the conversations she could not quite have with either daughter, the ones that required her to explain something that she herself could not explain, without also explaining that the world had organized itself around a lie so deeply embedded in its institutions that the lie had acquired the authority of fact. Ruth grew up knowing she was black. Her parents made sure of that because they understood what happened to black children who did not know it, who walked into white spaces without the armor of knowing who they were and what those spaces would do to them if the lineage was ever traced. But Ruth also grew up moving through a world that did not know it. She passed, not deliberately, not strategically, simply by existing, by walking into a room with her sandy hair and her pale skin and her freckles, and letting the world make its assumptions, which it always did, instantly, automatically, without asking. She found, as she got older, that she could not fully inhabit either world. In white spaces, she carried a secret that made everything around her feel slightly unreal, like a stage set she knew would collapse if anyone looked behind it. In black spaces, her skin marked her as other in the way that lighter skin has always marked its bearers within communities shaped by colorism, generating a different kind of distance, an accusation she could never quite disprove because it was written on her body. Clara, her older sister, took a different path entirely. She stayed in Birmingham through the civil rights years, marched, was arrested once on a bus in 1961, spent 2 nights in a county jail that smelled like chemical cleaner and old concrete. She did not carry Ruth's ambiguity. The world looked at her and made an instant, unambiguous assignment, and she had decided very early that she would live fully inside that assignment and push it as far as it would go. She became a school teacher, the same profession their mother had always wanted for herself, and she taught for 34 years in the same city that had put her and Ruth in different schools because of the melanin levels in their skin. They stayed close, these sisters, across every divergence their skin tones had created. They talked on the phone every Sunday. They gathered their children and grandchildren in the same rooms and watched the same reshuffling happen in the next generation that had happened in theirs. Some grandchildren landing dark, some landing light, the genetic lottery running its same and different mathematics through every new body born into the family. At one of these gatherings, in the late 1990s, a grandchild who had just taken a biology class in high school explained what she had learned about polygenic inheritance, how traits controlled by many genes at once produce a wide, continuous range of outcomes rather than fixed categories, how siblings can inherit very different combinations of the same genetic variants, how the same 2 parents can produce children who look, to the outside world, like they come from different families entirely. Clara and Ruth sat at the same table they had sat at as children and listened to a 15-year-old describe in the language of modern genetics what their lives had been, Clara said, at the end, quietly, that she wished someone had explained it to their mother that way, in those words, back when it would have made the walk to school slightly less lonely. Ruth did not say anything. She reached across the table and held her sister's hand. There are an estimated 378 genetic loci involved in determining skin color across humans and their nearest genetic relatives. Only 171 have been fully characterized. The other 207 have been mapped, but not yet fully understood. Scientists are still discovering the genetic architecture of the trait that divided these two women's lives. They are still finding genes, still tracing the evolutionary pressures that shape this spectrum, still building the full picture of what melanin variation actually is in biological terms. And what every new study confirms, with every new data set, is the same thing the data has confirmed at every stage of this research, that there is no natural boundary in the human melanin spectrum, no genetic cliff where one category ends and another begins, no place in the DNA where Clara stops and Ruth starts. The spectrum is continuous. It always has been. The boundaries were drawn by people, enforced by people, and they have cost families something that no scientific paper can fully measure, though some have tried. Edith died in 1979. She had lived long enough to see the legal architecture that had sent her daughters to different schools dismantled, though she had also lived long enough to understand that dismantling a law and dismantling what the law had built inside people were two different projects on two very different timelines. She kept a photograph on her dresser for the last 20 years of her life. In it, Clara and Ruth are sitting at the kitchen table, aged 9 and 7, eating breakfast. You can see, even in the black and white photograph, how different they look. You can see, if you know what you are looking at, the genetic lottery written in their faces. What you cannot see, because no photograph has ever been made that can capture it, is the weight their mother was already carrying at that table. The weight of knowing that the world outside the door had decided it understood what it was seeing. The weight of knowing that it did not. The weight of knowing that she would spend the rest of her life watching two daughters, born from the same love, carry that misunderstanding through every room they ever entered, and that there was nothing she could do to lift it from either one of them because it did not belong to her. It had been placed there by something much larger than any mother, and it would outlast her by decades. It still has not finished. What makes this story scientifically verifiable at every level is not just the genetics. It is the consistency between what the biology shows and what the data on human outcomes shows, and the absolute absence of any bridge between those two bodies of evidence that would justify what happened. The melanin system has no awareness of legal categories. The SLC24A5 gene does not know what country its carrier lives in. The MFSD12 pathway does not assign social meaning to its outputs. The genome does not know what Ruth was worth, or what Clara deserved, or what either of them should have been allowed to become. It simply shuffled. It produced outcomes. And then the world those outcomes arrived into decided, with the full force of institutional authority, that the shuffle result was destiny. The academic framework that social scientists call colorism, the systematic preference for lighter skin over darker skin within and across communities, has been documented across populations on 6 continents. It operates in hiring decisions, in criminal sentencing, in the allocation of parental attention within families, in the wages paid to immigrant workers, in the quality of medical care received by patients with different complexions, in the likelihood of a person's pain being believed by a physician, in the speed with which a bank approves a mortgage application, and in hundreds of other daily interactions that each seem small individually and collectively constitute a life. A 2006 study published in Social Science Research found that a 1-unit increase in skin darkness was associated with lower occupational prestige and lower personal income, controlling for education, parental background, and place of birth. A 2016 analysis of federal sentencing data found that among defendants convicted of identical crimes in identical circumstances, those perceived by evaluators as having darker skin received sentences averaging 8 months longer. 8 months for the same crime, based on the shade of a pigment. Clara knew this without reading the studies. She had learned it the way most knowledge of this kind is learned, through her own body moving through a world that communicated it to her relentlessly in small increments across decades, the way water shapes stone not through any single dramatic blow, but through the patient accumulation of ordinary contact. What she could not have known, and what would have perhaps given her some cold comfort, is that the science that now fully describes what her skin is and what it is not was being built at the same time she was living the consequences of what the world had decided it was. Researchers were sequencing genomes while she was teaching children in Birmingham. They were measuring melanin levels in populations across 4 continents while she was correcting spelling tests. They were discovering gene after gene with no knowledge of her or her sister or her mother or that photograph on the dresser. And what they kept finding, with every new tool and every larger data set, was the same result, delivered in the neutral, effectless language of published science. The human melanin spectrum is continuous, polygenic, and without natural categorical boundaries. Every legal system that ever drew a line across it drew that line in a place the biology does not recognize. Ruth outlived Clara by 11 years. She died in 2019 at the age of 73 in a city that looked very different from the Birmingham of 1953, and yet retained, in ways she knew precisely how to see, the same underlying architecture rearranged into new forms. At her funeral, her son gave a eulogy that included a detail Ruth had told him only in the last years of her life, something she had carried for most of her adult life without saying. She told him that every single day of her adult life, without exception, she had thought about Clara at least once. Not remembered her in the ordinary way that people remember siblings they loved, thought about what it meant that their lives had diverged the way they had, specifically, entirely, because of their skin. She had thought about the walk to school. She had thought about the 2 different routes. She had thought about the water fountains and the lunch boxes and the way white women on the street had looked at their mother with that complicated expression. She had thought about all of it, every day for 66 years. And she had never once been able to make it make sense, because it did not make sense, because it was never built on sense, because the thing it was built on was not biology at all, but the decision of a society made at a specific moment in history by people with specific interests in maintaining specific arrangements to treat a continuous biological spectrum as a binary legal fact. The genome never agreed to that arrangement. It has been quietly disproving it in every birth, in every reshuffled hand of melanin variants, in every pair of siblings who look like strangers to the world outside their front door, for as long as human beings have been having children. Edith's daughters were not an anomaly. They were not an exception. They were the ordinary result of the ordinary mathematics of polygenic inheritance, producing, as it always does, an outcome that does not fit inside any category simple enough to put in a law, an outcome that is, in the end, just a person carrying more melanin than her sister, or less, the way she carries every other genetic variant she received at conception. Neither more nor less than that. And the fact that a government once saw these 2 women sitting at the same breakfast table, born to the same mother and father, and decided that this was enough information to organize 2 separate lives, 2 separate schools, 2 separate futures, 2 separate places in the social order, is not a fact about genetics. It is a fact about us. The science has done its part. The genes have been sequenced. The pathways have been mapped. The spectrum has been measured and described and confirmed in study after study, on dataset after dataset, by researchers on multiple continents working in multiple languages, all arriving at the same conclusion. There are no natural categories in the human melanin gradient. There never were. There's only a continuous range of outputs from a biological system that has been responding to ultraviolet radiation for 200,000 years, indifferent to everything, except whether its carrier survives long enough to pass those genes on to someone else. Everything else that was ever done with this trait, every law built on it, every family divided by it, every life redirected by it, every daughter sent down a different street to a different school carrying a different lunchbox while her mother watched from the doorway, was done by human beings who chose to look at a biological spectrum and call it a border. That choice was made. It was not inevitable. It was not written in the genome. And unlike the genes that produced Clara and Ruth, unlike the melanin that the world read as destiny and biology knows as variation, that choice can be unmade. It just has not been yet.